Markets and our co-host, Zach Small, Cap Research, we are very pleased you have joined us for our quarterly Life Sciences Investor Forum. Our next live presentation is from Nirvgen Pharma. Nirvgen Pharma is a clinical stage biotechnology company focused on developing innovative treatments for nervous system damage caused by injury or disease. Please note, you may submit questions for the presenter in the box to the left of the slides. You can also view a company's availability for one-on-one meetings by clicking Book a Meeting in the top toolbar. At this point, I'm very pleased to welcome Mike Kelly, President and Chief Executive Officer, and Bill Adams, Chief Financial Officer of Nirvgen Pharma, which trades on the OTCQB venture market under the symbol NGENF and on the TSXV under the symbol NGEN. Welcome back, Mike and Bill.
Thank you. Appreciate it. Good afternoon, everybody. As the announcer mentioned, we are a emerging biotech company based out of Vancouver, Canada, and we are focused on technologies to assist the body to enable nervous system repair. Obviously, today I'll be making some forward-looking statements. You can find our financial disclosure on this slide, and our risk factors are listed on cedar.com. A couple key highlights about our business. The first is our technology. Many of you heard me talk about our technology and the breadth and the depth of preclinical data that we have with NVG-291, and that's really the cornerstone of the highlights of Nirvgen. If you focus on the right-hand side of this slide, our biggest news event is coming second quarter of this year. We will be having a readout of the chronic cohort of a spinal cord injury trial to see if NVG-291 translates from preclinical to clinical, and I'll talk quite a bit about that today. We also have an announcement coming up later on this month with our next program, NVG-300, which will have a preclinical readout in animal models of ischemic stroke, ALS, and a confirmatory study in spinal cord injury at the end of this month. Our company was founded by Dr. Harold Punnett. Dr. Punnett had a tragedy in his family. His daughter-in-law fell in 2016, and she was paralyzed. So Dr. Punnett, who's from Vancouver, went on a quest. His mission was to find technologies to help repair nervous system damage, to help his daughter-in-law get better. He came across the Nature paper, which is the middle of the middle picture on this slide. And the Nature paper explained all of the work that Dr. Jerry Silver had been doing since 1980 in his lab at Case Western Reserve. Dr. Jerry Silver, who recently passed, is a pioneer in nervous system repair. A lot of his works have been published throughout the decades, but NVG-291, our lead product candidate, was born in his lab. And basically what Jerry was studying at the time was when you cut your hand or you break your arm, your body tends to repair itself very well. But when you damage your spinal cord, your body protects itself and doesn't repair itself. So his work focused on, well, why is this? Why does the body not repair your central nervous system? He discovered that there's a group of molecules, chondroitin sulfate proteoglycans, CSPGs, that actually inhibit the repair. And then his second invention or second discovery was through a collaboration with Harvard, where they discovered PTP sigma, which is a receptor that CSPGs are attaching to signal into cells to inhibit this repair. All of this was summarized in his Nature paper that Harold Punnett found. And then Harold convinced Jerry and Case Western to in-license the technology to a group of investors in Canada, and Nirvgen was born. 5 years later, here we are 3 months away from proof of concept in spinal cord injury. So I talked a little bit about Jerry's discovery, but the most impressive thing about this technology, in my view, is the breadth and the depth of preclinical models where NVG-291R, which is the rodent version of NVG-291, has demonstrated effects. There's multiple models listed on this slide, ranging from spinal cord injury, stroke, multiple sclerosis, out to the optic nerve. It's the breadth of this this mechanism that allows this effect to be seen in multiple different models. And the most impressive is this is done inside Jerry's lab as well as multiple labs throughout the United States and throughout Europe. So it's pretty challenging when we saw the breadth and depth of this preclinical activity on where to focus. And the management team got together and we listed out all of the different disease indications where we felt comfortable going, and spinal cord injury was at the top of the list. Spinal cord injury rose to the top of the list for several reasons, not only based on the founding of the company that I talked about, but because of the impact of this disorder. It's cost $50 billion plus in the United States. It's also a very large impact to the Canadian healthcare system. And the depth of data that we have in spinal cord is spectacular, and I'll go through some of that. But it's also a pretty quick path to study clinically and a regulatory path because there's nothing out there for folks today. So what is spinal cord injury? This is typically an event that occurs. The average age of people who suffer a spinal cord injury is 43. It's mostly men. And they experience an accident, some type of vehicle accident, a fall, or sporting injury. And in an instant, their life is changed forever. They typically will have a crush of the spine, that they go into the hospital, they get surgery to decompress the wound. And then post-surgery, they do inpatient rehabilitation and then transfer to outpatient rehabilitation. But currently, there's no FDA-approved drugs to help sustain functional recovery. These folks will recover over time, and then that recovery plateaus around 9 to 12 months. It's important to note because that goes into our study design that I'll talk about in a few minutes. But after that plateau, wherever they get to, that's typically where they will be for the rest of their life. It's a devastating disease with multiple, with a significant morbidity. The cost is $1 to $4 million expected lifetime costs per person. This is a pretty wide range, but the range is due to the age and the severity of the injury. There's 18,000 people each year that sustain a spinal cord injury in the United States. There's a pool of 300,000 people living with spinal cord injury in the United States, and there's roughly 500,000 estimated cases in the world. We spoke to payers about this problem, and we asked them if we had a drug that could change or help these folks continue to get better, how would you value that? Most of those payers said to us, well, we would consider this an orphan indication, which we know that puts us into the hundreds of thousands of dollars per course of therapy. But then they said that they would give us a range. You know, they would expect that this if it offsets some of the $1 to $4 million, depending on the clinical profile of the product, this could be, you know, a multi-billion-dollar product opportunity in the United States alone. So I'm going to walk you through two different preclinical models when I said earlier that gave us confidence to go into spinal cord injury. And I picked these two because one of them focuses on the acute setting, which is meaning treatment starts shortly after the injury, and then the chronic setting, where treatment starts after that plateau occurs that I talked about before. So this is an acute model. This is where rodents had a spinal cord injury, a T8 or thoracic 8 compression injury, and the gray box shows you, or the blue box shows you that the treatment started shortly after the injury. The graph to the right shows you hind limb function. This is a BBD score, which is a standardized spinal cord injury preclinical model. Now, 21 is the highest that the rodents can score in function. And you can see after the injury, function falls off the table. There is some spontaneous recovery in the placebo group back to occasional coordination. But the treated group with NVG-291R got back to fine motor movements. So what does this mean? What does this look like? This is a video that is going to show two rodents. The one on the left is treated with placebo. The one on the right is treated with active NVG-291R. So you can see the one on the left, actually, there is no connection between the motor cortex of the brain down through the hind limbs. It's having trouble staying on the beam and walking, and the tail is down. Whereas the one on the right, there's absolutely reconnections being made, and the animal walks across the beam and turns on its own. This is a little bit clearer to see. This is a video, same study, different animals. The one on the left, there's obviously no connections being made. And the one on the right, you can see that there absolutely are reconnections being made. So this gave us extreme confidence that we should go into spinal cord injury in humans. And then we're looking at here is a chronic model. So you can see the gray box, the treatment was delayed 12 weeks. I mentioned earlier that humans plateau 9 to 12 months out after injury. Animals actually plateau at 12 weeks, and you can see that clearly demonstrated here in the light blue bars. You have some spontaneous recovery, and then it's flatlined and plateaued in the placebo group. Compared to the treated group, this is a C2 lateral hemisection. It's a different study done by Dr. Milton, but it's the same concept. It's showing that if the animals were dosed even after the plateau started, continuous improvement occurred. So you can see the improvement started within early as 4 weeks, and it continued even upon stopping of treatment at 24 weeks. So this gave us confidence that we could test this product both in the subacute stage as well as the chronic stage, which is what our clinical trials currently designed to do. We completed a phase 1 study. It's a single and multiple ascending dose, 70 patients. This drug is well tolerated. There were no treatment discontinuations. The most common adverse event was injection site reaction. So this is a peptide. It's delivered subcutaneously, just like insulin is a peptide delivered in the belly fat subcutaneously. And it's once a day. So we deliver it once a day in our trials for up to 12 weeks. The most common adverse event was injection site reaction. So this is the big trial that we started about a year and a half, a little over a year ago. We are working with Shirley Ryan Ability Labs, which is the #1 rehab hospital in the world. Wings for Life is a foundation that has helped offset some of the costs, about a third of the cost for this clinical trial. So we appreciate their support. But it's a single center study, and it's a 16-week study, 12 weeks of treatment, followed by 4 weeks of observation without treatment to see if we can see the continuous improvement that we saw in the animal models that I showed you. As I mentioned earlier, subjects are getting daily subcutaneous injections once a day for 12 weeks. There are 2 arms, 2 cohorts, a subacute cohort where people have been injured and then started treatment 20 to 90 days post-injury, and a chronic cohort where people start into the study between 1 and 10 years post-injury. The 1 to 10 years post-injury chronic cohort completed enrollment last December, and that is what we expect in the end of May, early June timeframe to see a readout of how those patients did. It's a double-blind randomized placebo-controlled trial, so we don't know that the study is still in the blind stage, but we will know shortly if this translates into humans. A little bit more about the design and the entry criteria. One of the key aspects of this is a traumatic spinal cord injury that we're studying, a C7 or cervical 7 injury or higher. And most importantly, these patients need to be motor incomplete. And what I mean by motor incomplete is they have to have some residual sensation or function or movement below the level of injury. So there is some residual connectivity going from the motor cortex of the brain down past the site of injury to the limbs. We were measuring motor evoked potential in these subjects, and what motor evoked potential is the actual electric signal going from the brain past the site of injury to sensors that are on the hand and the leg. They have to have a baseline signal in order to get into the trial. And why is that important? Because the primary objective of this study is to measure the improvement of that motor evoked potential over time. So subjects are entered into the study. They have a baseline motor evoked potential or a baseline signal that is going from the brain to sensors on the hand and the leg. And then they're stratified into drug or treatment, and they go through a rigorous exercise routine 5 days a week at Shirley Ryan. And the reason they're going through this exercise routine is if we can show connectivity, which is the primary endpoint, we're hoping that that connectivity will turn into functional improvement. So the primary endpoint is connectivity by measuring electric connections. And then all of the secondary endpoints are functional. Functional improvement. Does that connectivity turn into functional improvement? And so the first several that are on this slide here, 10-meter walk time, 9-hole peg test, and pinch dynametry, as well as the fourth, grasp. These are all performance scores. So we wouldn't expect to see any change from placebo or any change from baseline in the placebo group, but we would expect to see continuous improvement in performance like we saw in the animal models over the course of the therapy. So we're being measured at baseline, week 2, week 4, week 8, and week 12 at the end of the study. We're also measuring out to 16 to see if that functional improvement continues. So those are the primary and secondary endpoints. We have a whole host of exploratory endpoints in the clinical trial, from spasticity to sexual function to bladder function, to see if we can capture how this drug is actually working in humans who have suffered a spinal cord injury. And if all of those endpoints weren't enough, we are actually also conducting interviews, stratified qualitative interviews with subjects based on their experience, both treated and placebo and active patients are being interviewed. This aligns with the patient-focused drug development guidance from the FDA. It's been used by other companies, mainly to pick up if there's anything that we didn't collect in our endpoints that these folks may have benefited from. An example would be, I was able to get out of bed by myself. I was able to operate my wheelchair by myself. Things that we're not measuring in the primary and secondary endpoints. There's also some other companies working in this space. We often get asked who they are and what they're doing. The two largest ones are AbbVie and Mitsubishi Tanabe, are looking at protein inhibitors. Both are working in the acute model, meaning immediately after injury. Lineage, as many folks know, they're working on stem cell therapy. All of these different therapies are synergistic to our treatment. We don't consider these folks competitors. We're hoping that they win and they succeed the same way we succeed, because we believe that our product and their product eventually could have a synergistic effect over time. I touched on the first slide, NVG 300, which is our next development program. This program is actually in preclinical development at this stage. We haven't talked a lot about this molecule. We will announce at the end of this month, as I mentioned, the results of our preclinical studies that we've conducted. This molecule has actually demonstrated some very favorable pharmaceutical properties. It will be eligible for a biologic development path. And it'll have composition of matter protection beyond 2040. So we're very excited to have another program in our pipeline. So key highlights and upcoming milestones. So we ticked off the first one, initiating the dosing in the subacute cohort. We currently are enrolling in the subacute cohort. We announced about a month ago that the first subject was enrolled. Enrollment is going well. NVG 300, as I mentioned on the previous slide, will have a readout this month in preclinical animal models. And the big data is coming out at the end of May, early June timeframe in the chronic cohort. A little bit about our capital share structure. We have roughly 70 million shares outstanding. The announcer mentioned where we're traded on TSXV and the OTC, 21% inside ownership, and we have roughly $15 million in cash, which gets us way out beyond the chronic readout. And that's our presentation for today. So I'll open it up for questions and answers and ask Bill to be our MC of questioning. Bill?
Hey, Mike, thanks. Yeah, a few questions have come in here. First question is it a possibility that MDG-291 can be accelerated approval for the market?
Yeah, I often get asked that question. The FDA is going to have to answer that question, not us. We will ask. Obviously, there's nothing for these patients. And if this proof of concept study that we're currently conducting has the results that we're hoping it will, we think that there should be a discussion about accelerated approval. However, there's a lot of qualifications that go into that, and I'm not stating that we will, but I will state that we will ask.
Thanks. Next question, again related to 291, is our placebo group going to be offered this drug after the trial? Has the paperwork started with the FDA for the open label extension?
Yes, we will offer. We announced in a press release last year that we will offer placebo patients drug. There are a few things that we have to get done. First is we have to get approval from the FDA. We have to get approval from the IRB. We have a protocol that we will submit for FDA and IRB approval after this readout. So, we have to also make drug. We have some drug available, but, the three qualifications that we put in that press release is, number one, we have to see an effect in this study. Number two, we have to get FDA and IRB approval. But all during that process, we will be making more drug, and we will make it available to the folks in the placebo arm, yes.
A question on 300 is, will the data be released in a press release or in a presentation?
Likely in a press release right now. I don't believe there are medical presentations. There's no medical presentations within the next 3 weeks that I'm familiar with. We looked, but we will probably put it out in a press release and add some slides into our corporate presentation to kind of characterize what the effects were and what we did see.
Back to 291 again. Do we need to complete the study of the subacute before moving into a phase 3?
No, I don't believe we do. We've asked our regulatory consultants. We're working with the best of the best in the industry, and they are guiding us to, if you have positive data in this chronic cohort, have an end of phase 2 meeting, request an end of phase 2 meeting with the FDA, which is what our current plan is. So if we get this data end of May, early June, obviously that's going to be the top line view of the data. There's a ton of data to collect here and get it into a format that is digestible and presentable to the FDA. So we'll be targeting to get that meeting on the books as soon as possible and meet with the FDA, propose a phase 3 study at that meeting, and we'll see where to from there.
Great. Question back on 291. Are your plans to partner or develop and launch the product yourself?
Yeah, we are talking to a lot of potential partners. There's a lot of companies that are very interested in CNS, and a lot of companies that are very interested in this mechanism and they're watching to see if we're successful. Our goal is to get this product to patients as quickly as possible. And our intent is to take this product all the way and launch it ourselves. As you and I have talked, I've built commercial organizations that are much bigger than what we need here to launch this product. And we have all of the resources internally to conduct the phase 3. So the only resource we don't have is cash. And we will need some cash, obviously, to advance this program into phase 3. But our intent is to take it all the way. Now, with that being said, if there are bigger, more resourceful companies out there that can do this faster and get it to patients sooner, we're open to those conversations.
Great. A question has come in about other indications. Will this therapy help with those who have suffered brain injury via auto accidents and have now developed severe neuropathies?
Yeah, we have some preclinical data in traumatic brain injuries. Obviously, it's a very challenging area to do clinical trials in. But short answer is we don't know. If we get promising data here in chronic spinal cord injury and we get funded the way I think we will, we'll be looking at potentially other indications, all of which I showed on previous slide and then some. And traumatic brain injury, we do currently have some work being done in TBI.
We talked about partnering. The question related to, is there other sources of non-dilutive financing that we can look at? It's almost a question for me, but.
Yeah, I mean, we've done some non-dilutive funding with some small grants, small government grants. It's to get. Short answer is not to the level that we need in order to progress this in a phase 3. There's very few publicly government-funded phase 3 programs where, of the size of the phase 3 that we're going to need. But there are some non-dilutive fundings to do exploratory indication work, and that's what we're currently doing.
Thanks. The question on 300 again is NVG 300 based on Jerry Silver's work?
Yeah, we really haven't talked much about the mechanism of NVG 300, and I'm not going to talk about it at this presentation, mainly because we're still working on putting all of our patents in place, making sure that we have the data to support those patents as well. So I'm going to pass on that question at this time.
That's good. So the last question is what would success look like in the phase 1b/2a study?
Success to me would be if we hit our, we do what we wanted to do in our primary endpoint, meaning we show improved connectivity. So if we can show an improvement of connectivity, that shows that the drug is actually working the same way that it did in animals. That means that we're regenerating damaged axons. We're remyelinating damaged axons. We're enhancing plasticity. So if the signal becomes stronger, that is the first step, and that would be a scientific breakthrough alone in human studies. However, success would be to see that signal actually translate into functional trends. So, the study's not powered to hit our secondary endpoints, but like I said earlier, we're not expecting a placebo response. So, and these are a lot of performance scores. So every measurement time period, there should be improvement over the previous time period and continuous improvement, meaning there should be separation and continued separation. I know it's a long-winded answer, but the answer is really if we can see better connections and functional trends, to me, that would be a scientific breakthrough.
Great. I think we probably have time for about one more question. Mike just came in. Would a phase 3 trial be only in the U.S. or also in sites outside of the U.S.?
The goal would be to have both U.S. and outside the U.S. We know that it's definitely going to be U.S. and Canada. Right now, we're working to see if we would be able to open up European sites as well. But current thinking is definitely U.S. and Canada today. We have a little bit more work to do to see if we can open up sites in Canada, or I'm sorry, in Europe.
Okay. I mean,
so I think that was sorry, go. Was there any other questions, Bill?
I was going to say there's others coming in, but I think what I would recommend for people, if some of these other questions, if you want to arrange a one-on-one meeting, certainly there's an ability through the conference portal to do that, and we can follow up. So,
absolutely. In closing, thank you all for your time today. We really appreciate the support that our shareholders have given us since day one, and the continued support that they'll give us until we get this product on the market. It's an exciting time for the company. A huge milestone is less than 3 months away, and then the next big milestone is one step closer towards getting this technology onto market to help patients, which is our ultimate goal. Thank you all. Have a great day.